Welcome everyone to Precision NanoSystems Tea Time webinar series. So with the increase in genetic vaccines that's been taking place over the last year or so, and especially with COVID, we thought we'd take this opportunity to discuss some of the work that we've done and present some of our preclinical data. So Martin Rabel, who's our field application scientist based in Germany, will be talking about our RNA vaccine technology. So thank you so much for joining today. Just as a reminder of who your Precision NanoSystems teams are, you have myself, AJ, supported by Richard, who are based out of the UK, and then we have Jürgen and Martin, who are based out of Germany. So if you don't get an opportunity to ask us any questions today, please feel free to contact us outside of this webinar, and we'll be pleased to help. So I'll hand it over to Martin. Thanks, Martin.
Thanks, AJ, for your introduction, and also warm welcome from my side to today's webinar. And as said, I will be talking about Precision NanoSystems RNA vaccine technology. And as you all know, our vision is to accelerate the creation of transformative medicine that has a significant impact on the human well-being. Speaking about vaccines or speaking about pandemics, we all know that over the past 2 decades, new pandemic threats have emerged almost yearly with MERS, SARS, Zika, avian flu, and all things like that. And most importantly, the COVID-19 pandemic that influences us all. And the problem is that to bring a vaccine, not even on the market, but only to phase 2A of a clinical trial, pharmaceutical companies have to spend around $3.5 to $5 billion US. And when they have something on the market, the production time of these vaccines is still between 5 to 6 months to have a significant quantity of those conventional vaccines. If you ask yourself now, what is a conventional vaccine? Let me point you to that scheme. So on the left side here, we see what we would call a conventional vaccine. So basically a vaccine that contains the whole virus or parts of the virus in the form of proteins like subunit vaccines, or when it's a whole virus, live attenuated or whole inactive viruses. A better way to have a rapid pandemic response or producing vaccines for that is to look for an antigen like the small envelope glycoprotein, the spike clone protein, or the hemagglutinin from an influenza virus, and encode that into the genetic information, to a genetic code, and pack that into a synthetic carrier, a viral carrier, or transfect cells via physical methods with so-called nucleic acid vaccine. And this will be basically the theme of this talk. And as the synthetic carriers are the easiest to produce, best to modify, and also the best for a rapid response, we will focus on this synthetic carrier technology. First choice when working with these is basically based on a central dogma of molecular biology. So you can either use a DNA or an RNA-based vector. The benefit of using an RNA vector for your vaccine is definitely that you cut out one step in your molecular biology pathway. So when delivering the RNA, it can be directly translated in the protein or antigen of choice. Whereas when using a DNA, it first has to be replicated, transcribed, and then translated so that it's not so timely than with the RNA. And most importantly, also, there's a delivery challenge with DNA as it has to go to the cellular core. And the most used non-viral methods always deliver the genetic payload in the cytosol, and RNA is active in the cytosol. So we also, as PNI, choose RNA as the delivery vehicle of choice. And when working with RNA and want to produce a vaccine, you have 3 different base choices for RNA. You can either use unmodified RNAs, which is used by CureVac at the moment. You can use base modified, which was kindly pioneered by Moderna and BioNTech, and basically the 2 first vaccines that we have now for COVID pandemic are based on base modified mRNA. But today we will focus on the 3rd type, self-amplifying RNA, so-called saRNA, which is a technology that contains genes of the alpha virus that encode non-structural proteins that help to replicate this RNA and act as an adjuvant. And we have tested this technology in the collaboration with the Imperial College of London. So what is a self-amplifying RNA? Probably it's not known to everyone, and it is a bit newer technology. So as said, this type of RNA encodes for non-structural proteins that are responsible for making more copies of the said RNA, and thus more copies of the protein antigen result per molecule of the RNA. And how does that work? So first of all, out of the the structural proteins are produced, and the structural proteins basically are RNA-dependent RNA polymerases, RDRPs. When these RDRPs are produced, they replicate the same saRNA, and this is like a self-amplifying process. So they're generating multiple copies of the whole saRNA. And then from that saRNA, there are multiple subgenomic mRNAs produced. So a part of the RNA contains the antigen code, and this is replicated. And from this code, the protein is produced. So at the end, you have multiple copies of antigen with only one self-amplifying RNA molecule delivered into the cell. So the doses that you need to administer are lower compared to standard mRNA. But how to get there? So how to get a vaccine into a patient? So what are the steps? So the first step that you have to take is you have to sequence the viral genome. Then with bioinformatics, you have to build up an antigen library and select the antigens of choice. Then you have to manufacture those into an RNA, pack them into your delivery vehicle, which is a lipid nanoparticle in our case, and then you can administer them after manufacturing into your patient. And this seems to be still a complex pathway. Nevertheless, when we look at Moderna's case for the current COVID pandemic, we see that they were able to really produce clinical trial material 45 days, so not even 2 months after the first sequencing of the genome was done. And after 66 days, so roughly 2 months, they were administering these trial material into the first patient, which shows that it works. And as said already, 12% of the COVID vaccines that are now in clinical trials will be RNA-based vaccines. And the first 2 vaccines, so the front runners, were mRNA-based vaccines that were approved by the FDA. So it's a technology that helps you with speed of development, with a good safety, with manufacturability from low scales all the way to the big scale. The cost per dose is acceptable. And most importantly, you can encode any antigen. So when there's a mutation, it's really easy to incorporate this change in your RNA molecule and then deliver that RNA molecule with minor changes in the manufacturing process. So we learned now why RNA vaccines is the prime technology for pandemic response. But what is the vaccine workflow? So what things you have to tick off in which stages of your nanomedicine development? And we like to stage the nanomedicine development in 5 stages: discovery, early preclinical, late preclinical, clinical phase, and commercial phase. And the first thing, that's pretty obvious, you have to solve is antigen and target selection. So you have to look which antigen could help to promote a stable immune response. Then you have to select your vector. Our case, self-amplifying RNA. Then you have to take this and pack it into your non-viral delivery method, and the most used is a lipid nanoparticle. And this will start almost also in the beginning of the development, and the optimization of this carrier will be carried out till the late preclinical phase, just before the clinical phase. And then, to do that, you have to choose a manufacturing process or develop a manufacturing process. And this is something which evolves all the way from early preclinical phase into the commercial phase. And seeing that, it is important to choose a technology that can basically be used on all these scales and all these stages. And this is what we want to help you with our vaccine toolkit. So our vaccine toolkit consists of the genetic payload platform. So we can help you with sourcing RNA or even producing self-amplifying RNA. We also have our GenWay delivery platform, which is basically lipid mixes for different applications that can be used for the development of vaccine or not genetic medicine. And we combine these 2 expertises with our nanoassembler manufacturing technology, which allows the control of LNP formulation and influencing the physical chemical properties and biological activity with the manufacturing conditions. So let's start first. GenWay-LM is the delivery technology. So what is GenWay-LM? Basically, all what we call GenWay-LM will be lipid mixes that contain different lipids that will lead to LNP formulation when run on the nanoassembler platform. So usually 4 lipids are used: a cationic ionizable lipid, which is due for the interaction with the nucleic acid, cholesterol to mimic LDL and to give structure, then a helper lipid to prevent aggregation agglomeration during the production process, as well as the circulation of the body or after the injection, and the stabilizer, and a helper lipid to give structure and help with the endosomal escape and the uptake into the cells. So what's the difference? Why you should use an LNP compared to another nanoparticle technology, not a non-viral vector? Most importantly, these LNPs can exhibit multiple pathways to get into the cells and are therefore more efficient and be taken up into cells. So they can use receptor-mediated endocytosis and also other more passive endocytosis pathways. So it's easy for them to get into the cells. And then they have an intricate mechanism to be released from the endosome once taken up by having an interaction of the lipid components with the lipid bilayer of the endosome, and therefore releasing the payload into the cytosol, making it not only easy to get into the cell, but also releasing the payload. So GenYLM can be used for research use only applications, or most importantly, also we have clinical lipids, a suite of 73 different lipids that it can be used in the clinical stages. So we can be your end-to-end solution. So let us know what you plan to do, and we can help you with that. A huge benefit of LNP technology is shown on this slide. So what we see here on the left side is 3 different RNA types: siRNA, mRNA, and saRNA. And you see that they're really different in their sizes. Whereas siRNA, only a few base pairs, mRNA around 1 kilobases, and saRNA around 11 or even more kilobases. So a huge difference in the length of your vector, but still the hydrodynamic diameter and the PDI, so your physicochemical properties, are comparable, no matter, at least in this scales, no matter which type of RNA you use. So you can easily exchange your payload when you have a carrier in place. And also the encapsulation efficacy is not affected. So independently from the RNA type, you will be able to encapsulate over 90% of your precious payload, which will help with efficient delivery, a good packaging density, and also you don't lose any material, which means you spare some costs. So we thought taking our lipids into a vaccine model. And the first thing we wanted to do is we took multiple of our clinical lipids and screened them for their efficacy to produce an immune response against, in this case, an influenza model. So what have we done? So we took 5 animals per cohort. We dosed 2 times 1 microgram of RNA with a dose regime of 4 weeks apart. Bleeds were taken at week 4, just before the second administration, and at week 6, 2 weeks after the second administration. And the challenge with the virus, in this case with the influenza virus, was done at week 6. And what we can see on the left side is the hemagglutinin IgG titers in nanograms per milliliter after the 2 injections. In blue, we see Pabel. What is Pabel? We've done the studies in collaboration with Imperial College London. Pabel is a polycationic polymer that the Imperial College has designed and developed for vaccine usage, and that we have used because it's so well understood, and even the biological effects are really known, as a positive control. And what we see is, first of all, that all our LNPs exhibit a really higher immune response, or IgG titers, compared to the Pabel, and that with optimizing our structure from PNI IL-1VO2 all the way up to PNI IL-1VO1, we were able to have a stronger IgG titers, a stronger immune response, and we see levels of 10 to the power of 8, which is exceptionally high, and we even were not able to display 2 points because they were out of the detection limit. So really strong immune response. And we also checked after the challenge with the virus if it gives a protection, so if these antibodies are active. And what you can see here is the body weight of the mice after the challenge, and you see that with our LNPs, the body weight stays constant, so we can say that there's no infection. We now took this LNP, IL-1VO1, which was best, into a COVID model. And now it starts to get interesting, I would say. So first, again, we did the same. So 2 injections, 4 weeks apart, bleeds at week 4 and 6. But this time we did really a dose response study. So we dosed between 0.0001 microgram per injection all the way up to 1 microgram per injection. And again, mapped the SARS-CoV-2 specific IgG antibodies in nanograms per milliliter, compared on black to Pabel after intramuscular injection compared with intranasal administration. And we can see, first of all, a dose-dependent response. That's pretty obvious. The more you dose, the higher your levels are. but we see kind of a saturation effect with the 2 highest doses. But interestingly, also our really low doses, our nanogram doses, they still exhibit a higher antibody response or comparable antibody response compared to the payload positive control. To check that we not only have antibodies, but functional antibodies that are able to neutralize a SARS-CoV-2 cytotype virus, we developed, or the Imperial College developed, a neutralization test for us. And what you can see here are the IC50 values. And again, what we can see is that we have a dose-dependent response. The more we dose, the higher the neutralization effect. The highest doses, again, similar to the first data set, showed comparable effects. And we could have an IC50 of around 10 to the power 3 up to the 10 to the power 4 with our LNP technology compared to the payload control, where we see really low effects or almost no effects. And also the intranasal administration seems to give some protective effect, and intranasal administration would be really easy to really roll out also in and to administer into patients. Last but not least, we also checked, we all know that this is important for COVID, the T-cell response. So what you see here is basically, again, 2 dosages, but here after 6 weeks, we harvested the spleens of the mice, and then we quantified the IFN gamma splenocytes upon restimulation with SARS-CoV-2 peptides, so spike COVID peptides, and it is expressed as spot-forming units, SFUs, per 10 to the power 6 cells. And here we see that we have a strong T-cell response for the 3 highest concentrations, which really should give a good immunity after the vaccination. So now, after seeing that data, your question might be, but how can I produce these LNPs? Because these were like preclinical data, or small scale, several milliliter were produced, but you can use our nanosampler manufacturing technology. Why you can use that is basically because we have a microfluidic chip design that is incorporated in all our instruments, so in the full instrument suite. So from the microliter scale all the way to the liter commercial scale, we use the same microfluidic architecture, which allows for unparalleled control and reproducibility, and most importantly, also scalability of your formulation, coming from the preclinical scale into your large animal studies and tech transfer studies, all the way to a commercial production. And I want to just showcase that with an example data set. So what you can see here is a lipid nanoparticle formulation made out of Gen Y ILM, loaded with EPO encoded mRNA encoding for EPO. And again, left side encapsulation efficacy compared with the different instruments. So Ignite at 12 milliliters per minute production speed, Blaze at 60 or 90 milliliters per minute production speed, and GMP at 200 milliliters per minute production speed. And you see equally the encapsulation efficacy was above 90%. And also our physical chemical properties, size and PDI were comparable over all scales with minimal changes to your process. So still all around 75 nanometers, and PDI way below 0.1 for all these instrument types. So the nanosampler technology can really help you to overcome your key challenge you might observe right now with vaccine production or with nanoparticle formulations. So it gives you reproducibility. It gives you control over manufacturing conditions with an automated process that can be controlled by process or instrument parameters. You get high nucleic acid loading efficacy in a one-step process. So all our system produce the LNPs in one step. You get a rapid, effortless lipid nanoparticle production and optimization. Runs only take a few minutes, especially in the smaller instruments. And there's a seamless path to production. And to show you that, I brought with me, sorry for that, an example study from one of our clients, Genomics. And what they did, it is an siRNA-based platform, so it's a bit different here, but still they started their development on a benchtop, which came before the Ignite in October 2018. And they got with our technology into the clinic in April 2020. So like 1.5 years later, not even 1.5 years later, they were able to produce a GMP manufacturing run that can go into patients. So it really shows that the lipid nanoparticles combined with the mRNA platform and our manufacturing technology can help you for the next pandemic response or to rapidly bring your nanomedicines onto the market. And I think this sums up the data I have to present right now. So our genetic medicine toolkit is a versatile resource to streamline your development of vaccines, gene therapies, and even cell therapies by combining multiple expertises. And this was just a sneak preview of our vaccine development toolkit. And if you want to learn more, I would highly emphasize that you have a look at our NanoMedU training program. So NanoMedU is a training course that offers scientists introduction into the preclinical development of mRNA-based vaccine that we host once a month. So if you're interested in that, let us know. Leave us a comment, and we will be happy to enlist you in that. So thanks for your attention, and I'm happy to answer any question that there might is.
Well, thank you very much, Martin. That was a really interesting, very topical and informative presentation that you gave there. And thanks to everyone in the audience for joining us. We very much appreciate your joining us, interacting and asking all the questions. And finally, we will be running another one of these Tea Time webinars in a month. So do keep an eye and ear out. We will be sharing the details on social media and through our mailing lists. So we hope to see you in early April. Thanks everyone.
Thanks everyone.